When cells are exposed to sunlight, radiant energy can damage the DNA. For example, ultraviolet irradiation causes covalent bond formation between adjacent thymines on the same strand of DNA. When DNA is damaged in this way, it cannot be replicated or transcribed. Most cells, however, are able to repair damage to DNA. For example, an enzyme called photolyase can break the covalent bond between the thymine dimers. This enzyme requires visible light, which is used as the energy source for bond cleavage. Hence, this mechanism is referred to as light repair. A second repair mechanism involves an enzyme that excises the damaged segment from a single strand of DNA. This process is termed excision repair or dark repair and does not require visible light. The excised nucleotides are then replaced by DNA polymerase. DNA ligase forms the final phosphodiester bond.